let me go to the next speaker, who is very amazing and who is very well known in the field and developed a lot of algorithms. It's Valentine Svensson, currently working in Caltech. So thank you, Valentine, for coming. And you're
welcome. Yeah, thank you very much for inviting me to come here. I was very flattered by the invitation, and I'm enjoying the visit so far, at least. So as Nika said, I currently work at Caltech with Leo Pachter in the Pachter lab at the Department for Biology and Biological Engineering. And what I'm going to talk about today is mapping single-cell RNA-seq data to cell type taxonomies using cell type atlases as a reference. And it ties into the work of the Pachter lab. So Lior is pretty famous for making computational methods for inferring like splice variants and these kind of things in gene expression. And it kind of comes down to like properly handling the statistical inference of the things that you care about. So for example, in the case of splicing, you care about the isoforms which are expressed, and therefore you have to like have a statistical method to do that. So that, this like way of thinking that, okay, we should make an actual statistical method when we have a problem is why I wanted to work in Leor's lab on these kind of projects. I'll try to, no, sorry, I haven't practiced with this. Which one is, oh, okay. Okay, I'll have to figure this out while I'm speaking, sorry. So I'm just going to talk a little bit of single-cell RNA sequencing. There's been like a lot of introductions though, so I'll go very quickly through it. So I thought I would have to like explain a lot of things, but there's been lots of really good explanations from people who are really doing innovations in the field, like William this morning before lunch. And then I'm going to talk about something called generalized linear models. And it's kind of going to be a bit dry, but I'll try to not be too dry about it. And then I'll talk about like why those are useful when you want to annotate cell types which are in a taxonomic hierarchical structure, and how that can be useful once you have an atlas. So this is kind of forward-looking, like what would you do once you have an atlas kind of thing. So just talking about single-cell transcriptomic data. The idea is that you have a bunch of cells, and you want to measure the RNA in these cells, and then you somehow label which RNA comes from which cell, and then you count them. And that's the underlying structure of like how all of this works. There's just very many different ways of doing this labeling. So one way is like you take a bunch of cells and then you isolate them individually, and then you use like a mouth pipette to take one of them, put them into a tube, and then you do sequencing of the RNA in the tube, and that way you can like get this table which have like every cell has like different values for different genes. And that's like the data we're going to work on. And that way, like in one tube, you introduce one cell tag, and then you like look at the genes to see the sequences from the gene portion of these like hybrids, and then you get like quantitative values for each individual cell. Nowadays, we don't really do that. We use like flow cytometers that sort individual cells automatically into wells, into these huge like plates in a very parallelized way, which is usually automated using robots. And this will, then everything in one well in this plate will be labeled with one of these labels. Like one of them will have the label 6, and that means that this is the 6th cell, which means that you can tell from that which cell got which label, and then you see which genes were expressed in that cell. And then that's kind of like also a bit old style, but people are still doing it because it allows you to have a lot of control of your experiment. But there are, of course, recent developments where you are allowed a bit more control of the experiments, like the mini drop stuff that William talked about before lunch. But this is the idea of like you have these droplets, then that has been explained several times. You have like one like microparticle that has the label for the RNA, and then you have the RNA, and then combining these allows you to computationally split things up into this table. Something I'm very excited about is this new thing where you don't actually ever separate the cells by themselves. You kind of like take a bunch of cells, you randomly split them into like many small pools of cells, and then you label everything there individually. Then you pull everything together, and then you kind of randomly split this again. If you do this enough, and you do enough labelings in each of the intermediate steps, just by the statistics of like cells going together through this process, means that every cell will have an individual barcode. And I think this is like where we're heading, because it allows like really arbitrary scaling without a lot of like, there are also like issues, like you need a lot of input cells to do this, so it's not very good for like rare cells. Then you might want to do the flow sorting thing into the individual wells. But it's like, this is like a technology that will scale arbitrarily. So just like statistically, you will generate unique tags for each cell, and then you look at the genes, and then you can split this up into this kind of table, and then the data we'll be looking at in general would be like these cells, and every cell has all of the different expression values for each gene, and that way you can count these molecules. And now, the reason you want to do this is usually to do clustering, like was described recently, like or in the previous talk, that you kind of have a bunch of cells in your sample, and then you want to figure out, okay, are there like some types of cells which are pretty similar to each other, and like what genes are they expressing together? And then there's like about 100 or so different ways of taking your data and producing some kind of partitioning of it, so that you have like these different types. So here you would have like these like different cells, and you kind of like, based on the fact that these cells are expressing those genes together, you call that a cell type, and based on that you kind of can analyze that and like, you know, consider that one thing. This is an extremely difficult problem, and you have to like, there's really no solution. You kind of have to do it and see whether something makes sense, which makes this to like a half manual process. It's very like aided by many computational methods, but there's no like one size fits all method. You kind of have to like customize it to your problem and use a lot of biological knowledge to see like what was the assistance that you can use that is consistent with what we know about the biology of these cells. And yeah, that's like how to like make this like cell type annotations from your single-cell RNA-seq data. Why would you want to have cell types in general? Like, what's the point of taking a bunch of cells and segmenting them into types? And there are many like things. Like, if you can look for molecular marks for cell types, it means that you can like isolate them, and then you can like do other experiments further to see what they're doing, or you can try to like see what's different between one cell type and another, and see like, well, are they doing something that's like different from each other? Like, is this cell doing something in particular that this one is not doing, or does this cell have a capability that this one does not have? And what you can do when you have like an atlas of this like previously annotated types is that you can do a new experiment, for example, like these knockouts things, where you see like, oh, like I know based on the previous experiment which cells are which type, and now if I knock this gene out, does the proportions change in the types that I'm seeing? Is there any types missing in the new experiment? And of course, when we have the data, we want to say like, how certain are we that the things we're splitting up are like useful cell types? Like, do they have any functional meaning? So these are like just general uses for like having these kind of cell type clusters. So and this is like what most people want to do with single-cell RNA sequencing data. There's also like, of course, like the lineage stuff is very popular as well. But yeah, what we did was we looked through the literature a while ago to see like what people wanted from single-cell RNA-seq data, and it's like it wasn't completely clear. Like in the very beginning of the field, it was more about like, oh, there are these very precious rare cells, and we want to like study them. Like for example, 4 cell stage embryos from humans. Like you can't get so many of them, so you want to make sure that you analyze them completely. So you want to look at each of them independently, and that was like the main motivation at the beginning. But at some point, someone figured out that like, oh, actually, like we know about cell types, like we have known about cell types for hundreds of years, but can these RNA expression values like recapitulate what we know? And in like the 2013 or so, I think, or 2012, someone showed that this was like a very powerful way of reconstituting what we already knew, and also finding new things that we didn't know, like finding new cell types. And this was like so popular that it started driving technology into like larger and larger cell sample sizes. So you can detect more and more completely what cell types are there, so you're not missing anything. And you can detect rare cell types, which is very popular, because many rare cell types are super important. Like in the human blood system, there are these stem cells that live on for like 50 years in the blood, and they're just there in case you need to generate a new kind of like to repopulate your population of like different immune cells and these kind of things. So those are super rare, and to be able to find them, you need like to look at a lot of cells. So the technology started driving up to where you can like look at many cells and get a more complete picture. And this eventually grew to, for example, the Human Cell Atlas project, which figured like, oh, but we're doing this in the individual organs. Let's just combine all of the data to like one big atlas of all the types of cells which are in a human body. Or at the Allen Institute, which is a neuroscience institute, they've been doing this for 15 years or so, trying to catalog all types of neurons there are in a human brain, so that if you want to investigate this in the sense of like diseases, you can see like which neurons are affecting these diseases. Let's see. Right. So I made that figure like with regards to the technology development, because I thought it was like an interesting thing to see like how things have gone in the last 10 years. I went back to the data made to make this figure, and I updated it a little bit. This is like a less pretty version of the same plot, but I added some more publications as well. So this is just like the number of cells in the study, and then the publication date. And I wanted to see like, okay, given this, like how many cell types are there? So I looked at a bunch of these publications and saw like how many clusters or cell types are they reporting in their paper. And you can see a nice relation, which is that the more cells you're looking at in your study, the more cell types you're finding. And at least according to this, like this is not the best, linear fit, but there seems to be like a log linear relation here, which kind of indicates that for every 100 cells you sequence, you find a new cell type. And at some point, if we ends up like, there's like 30 trillion cells in the human body, once we sequence that, probably will like flatten out, I hope. But this is like actually going to make things kind of problematic when you want to analyze these things. So in the previous talk, Maria, they annotated like 50 cell types or something like this, and I'm sure that was like an enormous amount of work to go through all of these like marker genes, which ones are mentioned in literature, and you have to like do this pretty manually. Like, how do you even think about like so many categories? And just thinking about it, like in the very beginning, or in the beginning of this big explosion, the data sets were on the size of like 100 or so cells. Now we're in a state where like every single publication has 100 or more cell types, each supported by like hundreds of cells. So it's going to be like, and we were already back then complaining about how hard it is to deal with such big data sets. And now we have like data sets of data sets, which are these cell types and clusters. So like, how do we think about something, like how do we think about 800 cell types at once in a reasonable way? Like, do I use, so if I like want to say, okay, is this gene expressed in some cell types? And then you're going to get a list of, 400 cell types where it's expressed. Is that going to be useful to me? And this is like a big problem, like how do we use the data that we have? And it turns out that, so I'm going to introduce this slide a bit more, but in general, this problem has been a big problem in science for a long time, which is that, for example, there's a lot of species, and how do you group them, and how do you think about like do you have to think about every single species that exists when I talk about different animals, or if I talk about different bacteria, is it important exactly which clade of the tree they're in? If they're locally similar, the way vertebrates and invertebrates are like similar to each other in some ways. So the idea is to take some concept from taxonomy and try to do something with this. And I think this is a recent paper that is doing an atlas, so it's kind of about an atlas itself. The title is something about an atlas. It's published by the Linderson group. It was published in Cell recently with a backpack paper from another group. And it's basically making a taxonomic tree of the kinds of cells that they could find in the central nervous system of a mouse. And they made this taxonomy, which I'm going to use as an example throughout. But they made this taxonomy pretty much by hand. Like they have a very long method section where they describe why they put things together in this organization and why they think that makes sense and how they can validate that that is like a reasonable way of grouping things. But it's good because it has this like local grouping of things. You have neurons and non-neurons, and then among neurons you can like, split things apart depending on what you're talking about. Like, so if you are someone explicitly working on the spinal cord, you can like look down here and see what's the difference between these kind of cell types. But if you are working in a field that's like slightly different, you can like locally look at things which are very specific, but you only care roughly on larger distances between things. So this is like this taxonomy allows us to organize our knowledge, and that's what we want to use. Like when we have organized our knowledge, how do we relate other measurements to that knowledge? So the data, which is this like big taxonomy, yeah, I usually have slides with like notes with the numbers, but I think it's like 230 cell types below this like final taxonomic region, and at the final level there's like 27 or so like taxonomic branches. Let's see. Right. Here I have. So this data set, so I'm focusing just on the neurons branch of that taxonomy, and I'm going to use it like this. They're all from the 10x commercial technology of sequencing. There's about 75,000 total cells in this data set. They have annotated 214 cell type clusters, and in the last level of taxonomy, which is one level before what I'm showing here, there are 21 cell types. So this allows us to like think about the relation between these different cell types. Now, let's zoom in a little bit more to make an example, which is that, say that I have only like this annotation. There are 8 cell types. I have put the names here. I'm not going to like talk about them, but it's a good, you can refer back and forth if you want. And the idea is that, okay, I have this annotation of a bunch of cells, which I also have loads of measurements for. What is like, how do I think about those measurements in relation to like being a cerebellum neuron? Well, you can actually make statistical models for this. So that's this called like categorical probability distributions. So for example, like if you have something quantitative where you're measuring things and you can say like, okay, I have these 8 categories. Now, if I just look at a cell, what is the probability that this is a category 1 cell? And then you can have like, okay, I have written down that the probability for it being category 1 is pi 1. So say that that is like 10%. So if you look at a given cell and I ask you, is this a cerebellum neuron or not? If it's the 10% chance that it is a cerebellum neuron, you will say, no, it's not. So it's like not likely. And then if I tell you like, oh, but you know, out of all of the cells that I looked at, most of them were cerebellum neurons, like there were way more of them, then you will say, then you will like guess that it's more likely that it's a cerebellum neuron. And then you use that information about the proportions to inform your guess of what a given cell is. And that's just using the proportions. What you can do, so that's like basically say like, so when you are making that guess, like what a given cell is, you're basically weighing the odds of like that cell versus the other cells. Like what's the odds of seeing this cell compared to the other ones? Now, that goes into like this base rate here, which I call C. It's just like when I used sampled cells, this is how many I got from the different proportions. But there's other additional information. So say, okay, now I don't have my notes here, but okay, I don't know these genes by heart. But say like, if I show you a cell, and then I also tell you that this cell is expressed in NRGN, which is like a marker for one of these cell types, your guess should be influenced by that. Like it's not only the proportion, but like given the proportion, but also the fact that it's expressing this gene that you know should only be expressed at one of the types, that should influence like the odds of it being the thing. So it becomes like a conditional probability thing. So you have, okay, this cell is like, we don't see it very often, like it's a very rare cell, but look, it's expressing this gene and this other gene. Do you think it's this kind of cell, which are known to express these genes. And then you get a probability there, like I'm 75% sure that that is the kind of cell, but maybe it was just like random and we were unlucky, or something like this. And the idea behind this model is to put that there. So this X here would represent the expression of all of your genes, and this beta weight here gives you like how much you're influenced by a given gene's expression. Because I can also tell you like, okay, here's a cell, it's expressing beta-actin. Is that going to influence your decision? Because like beta-actin is expressed in every single cell. So it's not going to actually like be helpful in making the decision of like what cell type you guess this is. So this is like then what we call a general linear model, where we're making a linear model which like basically, okay, if I see the expression of this gene and this gene and this gene, if we add them together, that's a lot of evidence for it being this cell type. So that's like a linear combination of gene expressions that will give like a little value that tells you like how certain you are that it's a given cell type. And then you just plug that in, and then you like make a guess based on that, and then you evaluate like the probabilities of that with respect to like what was actually annotated in the data, and that way you can like find which weights are the best ones for the different cell types. So the good thing with this being a so like generalized linear models have some tools that come with them, which are going to be like really useful for different tasks that we want to do. So for example, if we want to find markers for different types, so for example, the type 1 here, cerebral neurons, it's marked by, I think it's IRS4, very clearly. Like, and what we do is we take these weights that we are using like as different amounts of evidence for guessing that these are different cell types. We normalize it by how common the cell type is, and that gives us an effect that's like specific to a given cell type. And the things which are going to like influence our guess to which cell type this is most will get high values in this thing. So this allows us to like directly see what are the things which are like marking these cell types straight from the data with one like given model. We can also see like, we can also make a comparison between cells. So say that I want to compare these cerebral neurons and the hindbrain neurons. What's the difference between them? You can also, again, use the weights from our generalized linear model to make what's called a contrast, which is normalizing both the ratios of seeing these things, like how common they are relative to each other, and we also normalize like every gene's evidence weight for what cell type this is. This allows us directly to see which genes are actually going to give us this difference. And this is like really powerful because we, like in the end, we just fitted one model, so we don't need to care too much about like multiple testing correction and these kind of things. And we can include like all of our data because this is pretty simple. We can also use this to predict cells. So say that you give me a new cell, and instead of like me guessing based on this different evidence, I have like trained all of these weights, and I have a computer. So the computer can predict which cell type this is most likely to be. And this is just showing like how well it's predicting. Like I leave things out and I predict based on the model, and it's like very accurate because yeah, it's pretty powerful. But this allows us to like, it allows us to like do two things, which is like both use it for new data, but if the data I already have is not predicted very well, it might be an indicator that our annotation of cell type might be a bit off. Or the way we're thinking about cell types of these cell types might not be completely related to the molecular measurements that we made. So for example, there's some confusion here. It's not a lot of confusion, but there's a bit confusion between 2, 3, and 4 compared to the other ones. So maybe there's like, it's a bit more fuzzy from the human annotation, which are these kinds of cell types. So it allows us to like evaluate these different things. So that's like the use of one of these models, which are nominal, which means that if we have a set number of categories, how can we like relate quantitative information to these categories? But we don't only have that, we have this hierarchical structure, which is like how I said before that we should be organizing information. And that brings in some complications, like and how to think about these models. Like we don't only want to work down in this layer. We want to use the fact that we know that there's like this cell type is this cell type. Like they are like, this is just like a different name for that, except this is in more general. So they are like the same in that regard. So when we as human annotators make this taxonomy, is there like a meaning that is related to the molecular measurements that we make that we can relate us to? So one thing we have to think about is like, what do we think about like this fact that, okay, all of these are central nervous system neurons, but they're all different from peripheral nervous system neurons. But like, what does it mean for this to be like more of this than this? Like there's like a degree of membership into like the different final layers. So there's like some commonalities between this and this, but they're not all common. So that's what we have to figure out a good way of dealing with that. So I'm just going to use as an example this little lineage here that goes down to this Cajal-Retzius cells. So it's like, this is like a bunch of cells. They are all Cajal-Retzius cells. They're all also di and mesencephalon excitatory neurons, which are a subcategory of di and mesencephalon neurons, which is a subcategory of CNS neurons, which is a neuron. So they're all neurons, but they're all like, different levels of neuron to some degrees. And then as we go down, like, what does it mean to to predict a cell being here as opposed to there? What does that mean? Or like, how much more evidence do we need? Because there's like a certain amount of evidence that tells us, yeah, this is definitely a level 3 thing. But then you get some more evidence and they're like, oh, actually, it's even more special. It's a level 4 cell. So we have to like think about this. And the way to think about it is that you take all of your different evidences that I described before. So this is your guesses, like, okay, based on this, you're like, so what I showed before, you're like taking every class and comparing it to the other classes. Here you're summing up classes together. So basically your evidence amounts. So you get more and more evidence for a certain class, and then when you reach a particular threshold, you can start calling something like a level 3 cell here. Like at some point, like there's enough genes being expressed in a cell that you're like, that's definitely like a 4 and not a 3. But 3 can be good enough sometimes if you just want to know like on a growth structure what things are. And in order to learn like how to do this, we want to share information between the different samples. So this allows us to do the same thing before. I should kind of explain the model. So the way this actually like works is that you keep the evidence the same for all of the genes across the categories, but you have a different offset for when you consider it to be enough evidence to call it a different thing. So these offsets for this evidence, so the evidence is a linear combination of expression values for different genes. So if you like increase the evidence by like, okay, this cell is expressing these 5 genes at pretty high levels, that's pretty high evidence, but is it high enough for me to consider it that cell? And these base rates for the different categories will actually give you when you have enough evidence to call something. And you can then like look at the effect size with respect to this beta, like which genes are marking the path down here to which things. So basically, when we are adding up evidence, if we add up a bunch of like these genes, we will end up in a place where we can call it like the final level 5 cell. And we can like again use this to predict cells, like, okay, if I look at a cell, is it going to be where in this hierarchy? And also this allows us to also look at like, okay, at what level of our clustering or like cell type assignments can we be pretty confident because we can actually make a prediction? And in this particular data set, it seems like the evidence for these cells being Cajal-Retzius cells is not super strong, because if I just like take the cells and I try to predict them back using all of the evidence that's there, it kind of cannot do it, except in some rare cases where it's misclassifying level 4 cells. Like, so it's yeah, this allows us to like maybe say, okay, I don't really trust anything below level 4. But let's at least look at level 4, because that's way better than looking at level 2. I will have a better idea of the cells which are in my sample. So it allows us to do these kind of things and like be quantitative about it. Now, the big problem here, of course, is that we don't have one trajectory, and now we have to like combine these two ideas of the nominal cell types and this hierarchical structure into like one thing that allows us to like share this information across these different branches. So we want to think about taxonomic cell types, and I'm just like looking at a sub tree here in order to like make it a bit easier to draw on the board. But yeah, now we're looking at these 15 types, and we want to like have a probability of generating a given cell type from all of these different kinds of things, annotations. Now, it turns out that there's actually like some graph theory that you can use for this, or what's called like partially ordered set theory, which is like a generalization of set theory. But basically, you have this collection of classes, which I've numbered here, and then you have like a structure of them, which induces a thing called a partial ordering. So we know that like 1 is ordered below 0, but there's no ordering between 1 and 2. They are like not comparable to each other, but we definitely know that like 2 is below 0. So what you can do with a structure like this is to partition it into something called anti-chains. So if you think about like this, you can look at like this, the sequence like 0, 1, 4, 10. That's like a chain because they have like a clear ordering between them. So there's like actually a unique way of taking a partially ordered set and partitioning it up into what's called anti-chains. So an anti-chain here is, for example, the set 3, 4, 5, 6, 7, 8, because there's no ordering between them. There's in no way you can compare 5 and 6. Like there's no like ordering. They're not better than each other, or they're like not below each other or above each other. They're just like not comparable. So that's like an anti-chain. So you take your partially ordered sets, you generate all anti-chains, and then there is like a natural order of anti-chains there. So then you have like an ordering between those. So what you get here is then that 0 is ordered above 1 and 2, which is one anti-chain, and then you have like this ordering. And then it turns out that it's pretty easy to like work with this structure once you have done this partition, because you can define an ordinal model between anti-chains. So that's this thing I described before, where we're just looking at the different degrees of membership down these different classes. And then within each anti-chain, you make a nominal model. So basically you have one set of weights for your genes that supply you with evidence of going down this tree, and then different weights for genes as to what you are within each part of the tree. And if you add together the weights, it actually gives you predictors for all of the classes in the entire tree. And it allows you to like look at the different weights of evidence for these different classes. And it also allows you to look at the base rates for generating this when you have the different proportions of cells which are in the different classes. So I'm just going to illustrate that kind of quickly. So you take your structure. This is the same structure as before. And then we just look at this set. And you partition it into these anti-chains which have this ordering. And then within each of these anti-chains, you make a model. So the thing to keep in mind here is that every red line or arrow corresponds to a different linear equation, which is like adding up weights for the different genes, which is going to be used as evidence for a cell being a type in this taxonomy. So, for example, if we want to compare cell types 1 and 2, we compare the ratios between them, and you have like a model for that. So these are the gene expressions. These are a vector of like how much evidence you put to different genes, and this is like a base rate of like how many of one of these cell types compared to the other are there. So that allows you to do that comparison. Now, you can also then make this kind of model where you add up the probabilities to go like down in this tree. You compare it to like not being at that level, and that gives you like a linear combination of weights to give evidence for that cell type. And then you can do the same for a different category. And that's, in the end, like, that allows you to define, so this is a way of like defining all models that you need. But just going back to the equation. So what we want to do in the end, we want to evaluate that probability. So say that we, instead of you saying y here, we take like a particular cell of interest, like yi or y1. We want to see like how likely is it that that cell comes from any of these categories. That means that you have to have an expression for each of these pies. But what I showed you before were about like ratios between different pies. So say that I want to evaluate this pi 6. So I have this cell. I want to say, what is the probability that this cell is a class 6 cell? Well, it turns out what you can do to actually uniquely find the, in a unique and meaningful way, the only values for these different parameters that are necessary is that you can kind of walk through this tree. So to get this value, you look at this ratio, which is basically here. So we put pi 3 as a reference category. We compare everything here to pi 3. And then we multiply that with the probability of ending up at this level in the tree. And if you multiply those two probabilities together, you end up with the probability that you care about. So you take those things, and then you plug in the different linear equations for this, and it turns out that you get like different, you can add up these beta parameters and these beta parameters, and that way you get the beta parameters, which is supplying you with evidence of being a category 6 cell in this thing. And similarly, if you want to like compare, for example, a category 6 cell with a category 2 cell, and I should just like point out that this is like a weird comparison, right? Because this is like a higher level up in the tree than this. Like if you just imagine the way it looked like, which means that you're comparing like a very broad category of cells to a more fine category of cells. But in this phrasing of this generalized linear model, this is a meaningful thing to do, which allows you to like contrast your specialized types of your like locally interesting cells to other cells in general. Like what's special about the cells that I care about, and what's locally special about different cells that I care about. And this allows you to like make an expression based on this, where you have to also normalize for the base rates, but that comes in here. You can also like use this. I get a new cell. This new cell, like where can I confidently say they are in this taxonomic tree? Then you might like predict that, oh, this cell C, I think this cell. We're very confident that it's like down here. Like that's pretty good. Like that's a good result, because we know very certainly which cell type they are in the final line of this hierarchy. You might get a result like this if you do this prediction, which means that we don't have enough evidence to say like which of this lowest level cell type it is, but at least we can say it's this one. Like at least we know it's a neuron and not an astrocyte. Like we know something about it. We might not know like what we might have known from another cell in a better experiment, but we know kind of what this cell is. And you also might end up in a situation where you predict this is just a cell, which means that like that's not a super good result, you might think. Like it just means that, oh, is this like we don't know anything about that cell? But that's kind of exciting, right? It means that you have now discovered a cell that's not in the cell taxonomy. So probably that means like, I have to like make another branch here. I discovered a completely new part of this tree. So it's a way of like thinking about it positively, and it's now exciting that it doesn't look like anything we've ever seen before. So that's like a way of thinking about it in a good way. But that's like what you can do with this taxonomy and like how to relate. So you have this, like imagine that it's 4 years from now and the human cell atlas has gone like really far and they have like 4 trillion cells in their database, and you make like a taxonomy that 400 people have been annotating for 2 years. Like now you have this tree, and then you make a new experiment, and then you can see like exactly where in that tree do you end up. So that's nice. And you can also like kind of evaluate the taxonomy by itself. Like, is it meaningful to have this branch at all? Should we just merge things together? You can just like, so this is like this will be very computationally intensive, but local changes in the taxonomy can be evaluated relatively quickly. You can just train like some different models, and then you can see, oh, actually like, if I remove that little branch, it turns out that my likelihood for my model is terrible, so I really need that little branch. And you can like then evaluate like small variations in the taxonomy to see like, oh, are we kind of like annotating things the correct way? Like, is this meaningful? So that's like another use of this kind of model when we have the data and when it's trained. So this software that implements this model is called Taxa, which is kind of Greek for like I ordered it. So it's implemented in TensorFlow, which is this machine learning framework. And we've been awarded this access to the TensorFlow Research Cloud, which is like a Google provided like free cloud thing, which uses these like cool TPUs, which are like graphics cards, but only for linear algebra. It's like really cool. And if you like nerdy stuff like technology and computers, those are like a really cool thing to get to work with. Currently, we have implemented a Nominal, Ordinal, Taxonomic generalized linear models in this framework. And the idea is to like make them servable so that you can use like query a cell, like upload a cell and then query where it is in the taxonomy and like train different models. We're using like a inference framework to learn all of these different beta weights that will easily scale to like billions of cells without any issue, particularly with these TPUs. So this is like how far we gotten with this. I don't have like any good like plots to show about like how it's running, but like the inference is going on, and it's like it's more interesting to look at the like how it would work. So, I mean, this is like my kind of conclusion slide then, that we can kind of, because there's such variety in like, and there's no one size fits all in this cell type clustering thing, it might be meaningful to use kind of treat it as human annotation, which is a bit noisy because people have different opinions, but if enough people do it with enough data sets, on average we can kind of figure out what's going on when we're saying that this is this kind of cell type. And then there could be a lot of arguments about agreeing about the names of these types, but yeah, that's a thing that's just going to have to happen. And the thing with this generalized linear models for the cell type is that we cannot relate the molecular measurements with what people mean. Like molecularly, like you know that this is this cell type and you know that this is the other cell type because of prior knowledge of these things. But in terms of the molecular measurements, what like do we mean something in particular, or do we have to like scrap the entire idea? Maybe we need like other kinds of data than the RNA data. Maybe we also need to put in methylation data or these kind of things. But it does allow you to like construct one single model for an entire taxonomy that can like basically combine all of the data together. And something I didn't talk about, but it's you can simply just like say whether the cell types are better explained by being from different labs versus being from different technologies. As far as, well, it kind of requires that more labs put in more data that have common annotations. But once you have done that, you can kind of like actually quantify how much of the cell type prediction comes from this being generated in this lab or it being from the molecular measurements. So you can easily like use that in one single model, so you don't have to like do a lot of normalization. You can use this because like with generalized linear models come this toolbox of like standard tasks, like prediction, which allows us to like look at new data in light of the entire data set that's been generated before. It allows us to look at the coefficients and investigate them, which means that we can look at markers. And it also allows us to compare cell types by looking at the contrasts in this generalized linear model. And of course, we can like look at the different trees by evaluating the likelihood of the entire model with the generalized linear model structure. So yeah, this is my acknowledgement slide. So I work here at BBE at Caltech. This is Leo Pachter, my supervisor. He's a big fan of this parrot that we don't actually have any legal rights to use, but he likes using it as a logo. We're kind of like trying to figure out if we can license it. So yeah, my funding is from this grant from the Brain Initiative to work together with Allen Brain Institute on a mini brain atlas. And recently we were awarded this TensorFlow Research Cloud, and I figured I should acknowledge them because it's pretty cool. Yeah, so again, thank you so much for inviting me to come here and talk about my linear model and cell types in general and single-cell RNA sequencing. So, yeah, so thank you.
And thank you so much for coming with all the obstacles you had. So, questions?
Hi, thanks for coming and thanks for your talk. I have a question about you actually applying this model to datasets. What is the largest dataset that you applied this model to? And I'm interested in this because I was asked the question, like, do I believe that that there are any cell type specific genes? The idea is that when you get a lot of cells, maybe you will start seeing that cell type specific genes disappear, and yeah, it's quite interesting to know if you're finding things like this.
Yeah, I mean, I could ask you the very first thing, like, so I've really only looked at this data set, and it's basically like, it's a pretty big data set, like relatively, but it's also like the most well annotated. So there has been like few more cell types which have like taxonomic annotations where you basically have 2 levels of taxonomy. But I think this one is interesting because the authors provided a 5-level or 6-level taxonomic tree, which is like a much deeper tree than the data set. So it allows us to like, okay, actually think about like these different relations. But we think that in the future there will be more levels in more data sets, and maybe we also have to like annotate some data ourselves, which is going to be a lot of work. And regarding the oh, actually I forgot your second part of the question a bit.
So the second part is do you believe that there are any cell type specific genes, like that there are genes that are expressed only in a single cell type?
No.
Is there any evidence to think so, or do you think otherwise?
So at least in the data I've looked at here, that is the case, but that might be like because that's how the authors are defining these cell types. So that's like a bit of a chicken and egg problem with these things. Like, yeah, all of what I talked about now is in relation to like people spending time to annotate data. The linear model itself does not presume this. It presumes that a linear combination of genes will identify a cell type. So you can have like this cell type is like high in this gene but not in this other gene, while this cell type is high in both genes but not in this third gene. So yeah, but the same genes are like expressed in both, but not at the same time, or not all three, or yeah, how to think about it.
I have a question. Have you tried other classification algorithms like decision forest or random forest or nearest neighbors?
Yeah, I haven't actually looked at it. That's on purpose because I want to have the interpretability of a linear model, because we want to be able to do things like these contrasts and to find the markers. So this is like by design. If I wouldn't do this, I would probably look at like neural networks because that's what everyone's doing. But yeah, like we just kind of want to make this like interpretable, and that's why we're trying this, because it allows us to reason about it in terms of these probabilities, which are like quantitative and nice. You can do similar thing with like support vector machines, but they're not as statistically robust. So.
Is the code on GitHub, or if not, do you have plans to put it to GitHub and to Docker container?
Yeah, definitely to GitHub. I usually just make Python packages which are relatively easy to install rather than Docker containers. But yeah, it is on GitHub in a private repository because I don't know. It's not my decision. I would just make it open, but it will be on GitHub as soon as we make a preprint.
Thank you very much. It was very intense and very interesting.
I'm sorry.
Do you plan to incorporate cell cycle information into your model?
I'm not like filtering out cell cycle genes, for example. So if you think so I've gotten that question before though. Like in this taxonomy, like if you have a cell type and the same cell type can be in a proliferative and a non-proliferative state, what do we do? I'm not completely sure actually. So I might like put it like annotate that as a different cell type because it will have like different things, and then it will share some things with the other one on the same level of taxonomy. But then you end up in a weird thing where like that is also like these other things, so you end up with multiple trees. But maybe that's fine. I haven't tried it, but it's something we need to think about.
Thank you.
So I think using a GLM to differentiate on the same level of taxonomy is not controversial, but using a GLM to differentiate between different anti-chains, but different levels, that seems a bit, let's say, not obvious to me that this would work. So what if you look at this model, if you look at the gene coefficients, which gene do you think would be predictive of on which level of taxonomy we are?
Yeah, so that will depend on, so the thing is, there is like, you can't necessarily look directly at the coefficients in the model. You have to look at the sum of them, which is going to end up, so in one way, you can think about this as like, maybe I should phrase it in a different way actually, but so we had this like another way of doing this would be to train, so say that we had 5 levels of taxonomy and then you have a bunch of different classes within one taxonomy. What if we like take one level of taxonomy and is make an ordinal, a nominal model in each of them and like ignore the other levels completely? That's like, and then you end up with these 5 different models. That's like one way of doing it, which is kind of fine, but you're then not sharing information between them. And what I'm doing is roughly equivalent to that, like learning these different individual weights on a per level thing, but you gain 2 things by doing these things. Like one is that you're sharing information, so if you have a rare cell type, it will share information with its like upper neighbor, which will have like more evidence for the different weights. So because you know everything for, say that you have like 200 Cahill cells and 5,000 of the one step above it, you know that everything that's true for the one above it should be true for the one below it, so that allows you to share information. So it gives you more stable weights for these like rare cell populations. Another thing it allows you is this outgroup, which is this top level, which basically means it doesn't look like anything, which you don't get if you're looking at like one level at a time, because everything might get like lower probabilities, like low probabilities in general, but in the end you would like you would only say like, oh, it's not similar to something, but it's most similar to something here. This allows you to actually get a value for it's not similar to anything, which is, I think it's very powerful.
Yep.
So you were working with rodent information, and I was wondering if you can predict cell types of human cells. Like, for example, there are many databases published, and they have some cells called unknown, right? So can you look at the gene expression profile in those cells, and based on this, predict what those cells are?
Yeah, I have some, like, on the very simple model in the beginning, so I've done some of those test analysis like that on the simple, like, nominal model. And there's, like, some stuff there where people have, like, basically annotated it as unknown or noise or whatever. And if you predict it from one data set to another, you're like, oh, actually, like, given here they annotated that as this cell type. So maybe it's not just noise. Maybe, like, there is enough signal to go between them. I haven't tried like a cross-species one, but I think that's like a really interesting way of doing this. And there is actually like a pretty recent data set that's doing a comparison, like from the Allen Institute, that's doing a comparison between mouse and human, and they're also relatively good. They have like a 3-tier hierarchy, so it might be interesting to like apply that here and see like, it also allows you to like check if things are real, because if it's nonsense between species, maybe it's like, well, maybe the species are completely different, but also like, we do expect some neurons to like have similar behavior and these kind of things.
Yeah, and also another question: do you need to look like how would you introduce the development in this, right? So you have adult animals and you have embryonic tissue. Can you predict the cells in embryonic tissue based on the information you have from adult tissue, or you need to do this kind of classification at different developmental stages?
Yeah, I would say this is kind of just thinking about terminally differentiated cell types, similar to the cell cycle thing, where you can have like cells can be in 2 states. They can also be like different degrees of differentiated, which allows like maybe you have to like make the tree have a depth as well, or maybe like within a level, like, so if I have a cell type, maybe I want to like split it up into like a categorical thing, or maybe like, I mean, actually that might be one way of doing it, like the same way that we have like the taxonomic level, and then within one chain we have that nominal model. Maybe like within one cell type we can have a continuous model of how differentiated it is. I haven't thought about that at all, but that might be like one way to go about it.
Thanks.
No. More questions? Okay. We thank you so much, and